Hi, everyone. It's nice to be here. I have clinic about half a day a week, so I just rushed over from clinic. But I think I'm supposed to tell an inspirational story, and I think I have one. I'll prepare you for the fact that what I'm about to show you by no means expresses the depth and breadth and capabilities of CCMB, but I think it's a nice story and example of how CCMB has really focused on one problem, solved it, innovated it, and done so in a way that's actually touching real inpatient care, and I think is really making a difference nationally and perhaps internationally. And I'll show you the story. So we're going to take a brief course into Crohn's disease and ulcerative colitis. These are inflammatory bowel diseases. I'm a gastroenterologist by training, but I have background in image analysis, biostatistics, and immunology, and some formal training there. So I'll tell you a little story here about how CCMB is really making an impact in these diseases. This is the only talk I've ever given where I've started with a quote, but I like this one because I think it's relevant. It's from Mr. Gates. And he says, "Without feedback from precise measurement, innovation is doomed to be rare and erratic, but with it, innovation becomes commonplace." And part of my research program in the clinical and translational space is really about measurement. How can we come up with better ways to measure diseases from a molecular perspective as well as from a macroscopic and even population perspective, and find ways to use data science to integrate that top to bottom, right? And precision medicine, which we've been talking about for 30 years now, is really only going to become a reality when we start making precision measurements. And I think that's one of the things that many of us are doing in all of our domains, in medicine as well as in other biologic sciences. So IBD, okay? So this is principally a condition that affects the colon. Maybe this is the wrong timing around lunch, but bear with me. This is a picture of what normal colon looks like, normal intestine. It should look similar to the palm of your hand, right? But these inflammatory bowel diseases are a condition of aggressive immunologic response, which we believe has to do with immune tolerance more than autoimmunity. And this is what your colon looks like when you have ulcerative colitis, where you have an aggressive immune response that ultimately damages the superficial mucosa. And we have a very deep understanding of this from a microbiologic standpoint, a genetic and transcriptional standpoint, as well as from an immune response standpoint. And there's a cousin condition, Crohn's disease, right? Which they're related, but the molecular functions are very different. This is typically more of a Th2 response, and the human level impact of that is much deeper penetration into the tissues, plus more systemic effects because of cytokine storms, as well as differential response to microbes. So for a patient, this can be as minor of, it gives me a bad day and I have certain food intolerances, or it can be completely life-altering, totally debilitating, and in some cases deadly, although fortunately that's much more rare. So this is what I've spent my life so far kind of working on. I'm 43, so hopefully I have a lot more time. But the way that we assess this, I show you pictures from a colonoscopy. Those are still images. And this is the primary means by which we assess these diseases, right? And for ulcer, and this is true for many different conditions where we use some kind of imaging as a primary macroscopic phenotypic assessment of the disease, right? This is the Mayo score, which was developed in 1979 at Mayo Clinic. And it takes ulcerative colitis and turns it into 4 degrees of severity. This is a Likert scale, normal, mild, moderate to severe. They put numbers on it so that they can claim it's quantitative, but it's really like just a categorical description, right? Frankly, this is pretty low tech, but this is the only thing that medicine's been able to do to come up with some kind of system that works and is reproducible. But we use this for everything. Staging the patient and phenotyping them, determining how much therapeutic response there is, giving them their prognosis, and probably most impactful is the fact that this is what regulators use, the FDA and the EMA. They use this as the primary means to figure out, is a drug effective or not? And increasingly, insurance companies and payers are using these scales to figure out, does a patient have access to a certain type of therapy? Will we continue paying for their biologic and small molecule therapy based on this score? But there's a lot to be desired from this, right? And there's a ton of problems with this. even when you have experts, there's an enormous amount of disagreement because, as all of you can understand, these are qualitative assessments. Like if we go back to these images, it's hard for me to tell, and I do this all day, what is the line between mild and moderate, right? And that concept can be whatever you want. So the ways that we've tried to address these bias problems, subjectivity problems, and expert disagreement are to spend a ton of money on what's called central reviewers. So we're supposed to send our colonoscopy videos to some unbiased, blinded human who allegedly is an expert at doing this. That's right. Literally blind in many, many ways. In many ways, but I'm trying to be nice. I'm trying to be nice. Pharma spends a fortune on this. Payers are now demanding that we do this, and I send my colonoscopy video to Blue Cross Blue Shield or United Healthcare for them to tell me how severe the patient is and make a major determination for the patient. So this simple score, this simple thing has enormous impact for the over 1 million patients in North America that have these conditions, not to mention the money. The money is a whole other story, but I'll just tell you, it's a lot. And our ways to try to solve this are just unfeasible and untenable. So here's the background. Here's the problem. This is my entry point in 2018. This is a little story of CCMB. This is my entry point to CCMB. And I have a little bit of knowledge on image analysis that's a story we won't get into now. But can we use data science? And just at this point, convolutional neural networks and other artificial neural networks are starting to become available. They're starting to begin to enter into the clinical domain, but there's still not a lot of expertise on the GPU access isn't there. So it's really small pockets of knowledge. The CCMB, of course, had that knowledge. They were way ahead already. For them, this was 2010 technology, and they're like, why are you bothering us with this? But I'm like, humor me, because I think that there's some legs here. So this is what CCMB is supposed to do, right? Take the clinical expertise with a little bit of translational science knowledge and combine it with the breadth and depth of technical and methodologic innovation that's possible in CCMB, bring these people together and really try to make impacts on maybe a small scale and in some cases a very large scale. So this was my entry point. I was nice enough to meet Kayvon. He's like, yeah, sure, Ryan, we'll figure this out. This is simple, but no problem. And it's turned into a great relationship and friendship that I'm incredibly grateful for. But a big team of people on both sides. So what do? We started simply, I'm going to blow through this part because this is old hat to everybody. But in 2016, 2017, 2018, neural networks, no one knew what I was talking about when I put my clinical hat on. And I'm like, you got to trust me. This is the way to go. So more or less, we pump a bunch of images in. We take a couple human reviewers. They take a year to manually annotate all these images for the severity. We train a CNN, no big deal in 2023, but then this was a big deal. And we came up with a model that could take still images and give us a predicted probability of what's the Mayo score here. So you've got some automated, standardized, non-biased method for grading disease. I'm not spending money. I don't have to worry about the expertise of the individual. We all know this now. This is old hat, but this is where we were then. And suffice it to say, we were able to show that our simple system based on 20,000 images, which runs, of course, very quickly, matched the agreement for image-to-image agreement of paired expert humans that are getting paid about $600 per colonoscopy video to review this. So from just a financial standpoint, this was huge. But we showed this. I was surprised this made a big impact because I didn't think that it was that big of a deal, but the field was really interested in this. But these were just still images. I could build the neural networks at this point. I could put the CNN together. And frankly, I'm just stealing code on GitHub. That was work, but that's how kind of coding is at this point. But we needed to go further. And this is where the knowledge and impact of all of us in this community really came to play. When we look at real world video and motion analysis is really a complicated thing. This is a still image from a colonoscopy. And you're like, oh, well, I don't know what that is because I'm not a doctor. You don't know what this is because this is nothing. This is the camera bumped up against the tissue. Most of the colonoscopy, most of medical video, in fact, a lot of CT, MR, endoscopy, laparoscopic video is noise, is useless. And just distinguishing noise from not noise is a huge problem. And so the next year, we said, how do we adjust for these real world distractors? right? And this now become more complicated. We did not use VHS, but more or less throw back to the old days. But we're getting increasingly sophisticated. And this is all, through Kayvon's lab and frankly, a lot of Kayvon's students who work very closely with me clinically, actually came and saw some colonoscopies. And we really had a nice exchange of knowledge here and developed multiple neural network modules that were now interacting together. Pre-processing modules, de-identification modules, and informative image detectors to figure out, is the image useful or not? Delete those images, figure out other distractors. We take a lot of biopsies, and those biopsies often look just like an ulcer. They look like disease, but how do you distinguish whether or not what you're looking at is iatrogenic impact or is actual disease? You have to build subsystems to do this. Pardon me? Iatrogenic. Yes. Oh, I'm sorry. Iatrogenic meaning that is the damage you see from something that the clinician did or the operation did, or is it native disease? Does that work, Gil? Yeah. Okay. So ultimately, we're making slow progress in 2019 here. Now we're finding ways to try to get these neural networks to talk to each other and come up with a whole video predictive endoscopic score. And this was a lot of work from Hemming Yao, who has graduated, but had a lot of hard work on this, and she led most of this effort. And ultimately, we were able to build a fully automated video analysis system for endoscopy, which nearly matched the review capability of paired central reviewers. So this was really doing the professional level expert work on full motion video, regardless of the source. So this led to a number of publications, and in fact, 2 of our publications got standalone editorials for the space because they were really recognizing that these small impacts in data science, and particularly image analysis, are making a huge impact in clinical medicine and really changing things. So everybody's excited about this in the clinical space. We're providing reliability, efficiency, and speed, lower cost, frankly, eliminating the cost, and giving uniform, non-biased measurements. Precision medicine, really, we're giving you the right kind of measurement, the same reproducible measurement all the time. The field is thrilled with this. Kayvon and I were not satisfied because we felt we could do better. If we think about these Mayo scores, and you can extrapolate this to lots of other medical conditions, it's 4 levels, right? But it's even simpler than this. The FDA and the EMA, they consider 0 and 1 to be the same. They consider 2 and 3 to be the same. And this type of construct is similar in oncology and many other disease states, transplant medicine, where they have to bend these conditions into very coarse measurements. But there's so much granularity and detail here that's being thrown out. We felt that Michigan had the opportunity to really innovate in this space and come up with new ways to measure disease. Think of these situations. Here's a patient, and this is a colonoscopy graphic where most of their colon is severe. Compare them to this patient where they also have severe disease in Mayo 3, but only a portion of the colon's involved. Are these 2 patients the same? Our current measurement says they're identical, but they're not. What about if you have 2 patients who have a therapeutic improvement on treatment, but the distribution changes? Are they the same? And what if you have a mixture of different disease compositions? And this may be severity grade, or it may be phenotype grade. This may be true at the macro level, but it also may be true at a histologic or even a multi-omics level. But you need to be able to parse out that granularity. So this is what we worked on. We're failing, the field has been failing for 40 years to really quantify this disease. And we thought with data science, we could really make an impact. But this part was going to be hard. We had to innovate the complete structure of how we look at this, and principally to be able to come up with a methodology using image analysis, other metadata, to try to get some kind of localization or geographic specification of where are we, more or less a spatial mapping of disease. And not just what the grade of disease is, but what are the different features of disease? Are there ulcers there? What's the histology at those segments? What are even the genetic and transcriptional signatures at those segments? So this is just a slide to basically say it's complicated, right? That's all we need to know, at least all I need to know. I did pretty well in mathematics, but I had a limit at discrete math. I can follow this, but we didn't write it. But we worked together on this methodology for macroscopic spatial information. And then translated this together where I worked with bioengineering here as well and brought them into the game to try to come up with in vivo sensors to give us location data for the patient. Now, it was coarse because the technology is not quite there yet for ultrasound and laser capture and RFID inside of patients. But what this is taking the new methodologies that we developed together, mapping the predicted path of the colon and then the actual path in blue. The red is predicted, the blue is the ground truth. And over several models, we got really close. And what ultimately this translated to is a completely new way to assess inflammatory bowel disease. We take endoscopic video and we perform multiple different levels of computer vision analysis. Frame level pre-trained neural networks for severity classification and disease features, and combining that with the positional location estimation, which is completely innovative and new. And really, there was nothing in the literature about this. So I can confidently say we invented something. And this is what we're using now. And we are now pitching this to the field, and we call it the cumulative disease score. So this is something real. It exists. It's out there. And in my small little world of gastroenterology, we're really making an impact. And we're spatially mapping. This is an example of spatially mapping the disease severity for the patient, but we can also overlay all kinds of other things. The histology, the omics from the biopsies that are collected, both transcriptional as well as protein, and even microbial signatures that can get mapped spatially so we can understand how these things interact, and all leveraging existing data science. So what has been the impact here? Pretty huge. We partner with Johnson & Johnson on live clinical trials with them. And what we've shown is that using our score, we're informing the entire clinical trials community that the buckets that they've put patients into for severity are really limited and their results really need to change. So in clinical trials, these are Mayo scores, normal, mild, moderate to severe. And in a clinical trial, you assume if you have a Mayo 2 or 3 that all those patients are the same. They all go in the same bucket of being active. These patients are inactive. And what we're able to show them is all these patients that are screening into your trial, they have a huge mixture of the amount and severity of total disease in the colon on a per patient level. So in this bucket, there's a spectrum of patients. Sure, so these are traditionally severe patients, Mayo 3. But this red here in that bucket, sure, about 40% of these patients match up with our score as being severe. But look at the distribution. There's so much heterogeneity in this population, and this has a lot of impact for drug development. So we're replacing conventional scores like these Mayo 2s and 3s of severity with our cumulative disease score, which is giving so much higher resolution and informing drug development at a far earlier phase. They're seeing signals now in phase 1, 20, 30 patients that they could never see before because data science is providing the granularity. New metrics for IBD, it's completely quantitative, it's explainable and traceable. Getting a lot of, some of Brian's comments on generative AI and some of the needs for traceability, trustworthiness, evidence, we can provide that. And we're capturing more of the physician's assessment. When we show this to the doctor that performed the colonoscopy, they have no questions. They're like, that's exactly what I saw. So this scans well with the clinical experience. This is some of the impact that we're having in clinical trials. This is, as I'm sure you all know, this is just a power calculation, a power plot done for the purpose of sample size estimation in clinical trials. Using traditional Mayo scores, this is your power curve that we've had for 30 years. Using the new score that we developed here, you gain so much power that in multiple randomized controlled trials, we've shown with our score, you can detect the same degree of clinical and therapeutic difference with half of the patients. Cut the cost of the trial in half. Speed up drug development by years, right? And also get a go, no-go decision on if your asset is worthwhile very early. But we're even moving past that, taking this type of CDS data, and this is some work from Christian, where we're taking this data, inputting it with clinical metadata, and actually from the screening colonoscopy, predicting what is your likelihood in 52 weeks of having a therapeutic response before the drug goes in the patient. You can actually tell prospective subjects based on your characteristics, this is your pretest probability of having a response to this drug. And if you go on placebo, we can figure that out even more. And then we're implementing this real world. So what excites me the most is that these data science innovations, with all the work together in CCMB, we're able to push this out to touch real patients. Shown you clinical trials, but we're actually doing this in clinical practice now. So at all of our endoscopy sites with the University of Michigan, we actually have something called Access, which is our automated Crohn's and colitis scoring system, where we are passively, with no clinician touch, capturing all this endoscopic video, pushing it to a server that sits in Domino Farms. And this is just running in the background all the time. So this objective, standardized, reproducible data is available to clinicians right now. So we're really solving a lot of problems in IBD. We're ultimately, by taking a lot of innovations in image analysis, combining that with new types of modeling, such as fuzzy neural networks, we're really getting better answers for patients and trying to predict things in the future, because we're doing more precise measurements that match up with the patient. So I think that this is a small story, but a good kind of inspirational kickoff story for CCMB, because I think we've got the right people here. We've got the perfect blend of clinical and translational scientists with innovative methodologists and that span a spectrum of biologic sciences, data sciences, and computational medicine. And when you take that plus all the tools that we have at Michigan that's really special, I think that we're finding the right solutions. So it's been great to talk to you all. And I hope you all have great stories like this to provide next year. So.
Ryan, that was fantastic. And I hasten to say that I bet it will be memorable for this entire audience. Thank you so much. And is it okay if we put the recording in the website? Margaret.
I can come to you. It's like Donahue. It's showing I'm old, right?
Yeah. Welcome, Ryan. No, I don't really have a detailed questions. I just have a comment for, since we are having a CCMB crowd here, is I think your talk illustrated why we went from one MSTP student every 2 to 4 years to now having 4 or 5 MSTP students in the program. We used to be not so popular for MSTP because we don't say, oh, all the classes you took in medical school are similar to what we are teaching, therefore you can go straight into the work and don't have to take classes, which is what CMB and human genetics and many of the other programs do. We asked them to do programming, linear algebra, all of these good things that are nothing like what they are taught in medical school. But we now have a pretty good class of MSTP students who are interested at exactly that interface and are taking the same classes as our students and are doing well in the program. I think one of them is also called Ryan. So I think that was just a comment. Thank you.
Great. Chuck, go ahead.
So I understand why this is going to be really important for drug development and things like that. But I still am missing the clinician getting the information and choosing a therapy based on this. Are we there yet?
Yeah. Well, I think so. Clinician, I don't think that these types of...
Hold it up. Just hold it up.
This works? Okay. All right. Thank you both. I don't think that we're giving the clinician any necessarily huge insights. What we're doing for them is we are providing a quantitative means to justify their intuition in most situations. So in our specialty, we don't have the luxury of most of our patients having a complete response going from severe disease to totally normal. So it's always some kind of increment. And so the clinician is really interested in, I appreciate there's been an incremental decline, but hasn't been enough. Am I biased in imagining things because I want to continue treating the patient? And I think that the clinicians are all very aware of this. But if we can arm them with quantitative data to substantiate their decision making, I think that's one arm of this. But then the other arm is, much like art, we don't always understand why something is or is not art. And we don't always understand, at least myself, why I think I should continue a patient on a therapy or why should we should change. And so this type of analytic output can help us understand more what is the phenotype or the quantitative description of patients that correlate with one who will have a positive course versus one who will have a negative course. We're still learning because when you make a new metric, if you're a proper methodologist, it takes a lot of rigorous work and time to really understand if these are useful or not. But we think it will be, and certainly the field's excited. But I appreciate your point, and it's spot on.
Follow-up question. So I'm in oncology, and I totally like this is really powerful. So I'm a huge fan of higher resolution data. You have higher spatial resolution at multiple levels. So I love the work. One of the things that we run into in oncology, just with the clinicians, and that's why I'm really interested to hear what you see in GI, whether it's different or the same, is that we'll have new metrics, we'll have new tools, and we'll say, hey, this can actually give you a better, more precise, right, understanding. In this case, it's like we're predicting infection in immunocompromised patients. And then I get two responses. So there's one group of people who's like, oh yeah, that makes total sense. Yeah, I would like to know. Yeah, there's a probability there. Yeah, I'll integrate it into my day-to-day clinical work. And there's another group of people, and I'm not judging either of them, but this is just observation that says, well, yeah, that's probably good, but where's the prospective randomized controlled trial that proves that this improves the outcomes and all of that? And I'm just sort of curious of what your experience has been in the response of clinicians to the tool.
Yeah, so I think there's two brief ways to answer that question because I don't want to take too much time. One of them is granularity does not ensure that you make any better of a decision. So when you apply these types of tools, the quantitation is attractive, but it might not matter. So something that we're doing more of is applying decision curve analysis to try to look at, yes, you have higher resolution data or more quantitation, but how much of that information is actually impacting your decision? If I'm going to send this patient to surgery regardless, and it doesn't matter what I see, then I don't even need a video. Like I can know nothing and guess surgery all the time for this kind of patient. And my guess is as good as all this quantitation in the last 5 years of Kayvon and I's life. So I think that it's really important, and increasingly in the clinical journals that work with AI and ML, we want to see decision curve analyses. And this is actually something that Vickers and Karen Deep Singh have introduced me to, which I think is a really useful way to look at it. But then the other part of it is with the RCT. The challenge with the RCTs are that so much of our decision making is already baked in before you get to these measures that we're finding it increasingly difficult to tease out those uncaptured nudges that push the clinician behaviorally to do one thing versus another. So I think we just have so much work to do. People are scared of generative AI. I'm not worried about it because we're going to have so much work to do in figuring out how to analyze it and how to properly report it so that we know these things are useful or not.
Yeah, no, I totally agree. I think the RCTs are totally impractical for a lot of these things. So I was just sort of curious, like how you're handling it. So thanks for that.
I'm grateful for the FDA though, because it's kind of like pumping the brakes so that we have some time to all catch up. And I think they're stalling with the RCTs in not the SMAD space, but
RCT is.
Oh, randomized controlled trials. Randomized controlled trials.
Okay, listen, this is great. Thank you very much. Thanks for the comments and questions. Let's now turn.